OSIRIX. So welcome to another episode of Science of the Times. I'm Simon Khalid.
And I'm Tim Colson.
So Tim, how's the week been?
Oh, it's the end of term, so holidays are in sight. Heading off to South Africa in less than 2 weeks. But how about you?
I mean, I thought every day was a holiday for you, but we won't go into that now. Okay, thanks. I'm really excited about today's guest because it's, I'm a bit biased. It's quite close to my own research. So
no, and he's in my department. So,
yeah.
we both know Tom. And it's all about the sort of chemistry of the way that life works and the way that cells operate.
Yeah, absolutely. It's all about sort of possibly understanding why biology chooses molecules to be arranged in a certain way over others. When there's a 50-50 choice, ostensibly anyway, a choice.
Life is selective of the shapes of molecules.
Yeah, the way shapes are arranged in space. So it turns out that he might well have cracked this problem that's been plaguing people for a long time.
No, that's super exciting. Well, before we invite Tom Richards on, just to say, please, if you enjoy the podcast, please subscribe. We'll put a link to Tom's paper that we're going to be talking about in beneath the podcast. Also, please like it and please share it online.
Yeah, tell all your friends all about it.
Tom, would you like to introduce yourself?
Okay, so my name's Tom Richards. I work in the Department of Biology at the University of Oxford, and my research team is really interested in understanding the diversity of microbial life. And what we realised quite early on, along with many other people, is that actually most of the tree of life is composed of microbial organisms.
And that's not just bacteria and archaea, is it? No. There's all sorts of things.
Indeed, a lot of our close relatives are actually single-cell microbes, but like us, they also have a nucleus where they store most of their DNA, and that's the group we're really interested in.
So those are eukaryotes.
They are eukaryotes, yeah. And so we there's a specific type of eukaryote that's sometimes called protists, but we also study microbial fungi, and we're trying to fit them on the tree of life and then understand how the tree of life was shaped by various evolutionary processes.
Well, our listeners know all about the tree of life now, don't they, Tim?
I was going to say exactly that. So, because Simon and I chatted to Max Telford about the animal part of the tree of life. So it's fantastic to actually get you on, Tom, and hear about what most people is kind of the overlooked bit. But before, I mean, you've kind of possibly got a solution to one of something that's been puzzling biologists for a very long time. We'll come on to that in a sec. And that's Simon's the real expert questioner on this, the expert questioner on this compared to me. But before we get on, you told me something amazing just before we came on air, that you've discovered a single-celled fungus that's got an eye.
Yes, yeah. Actually, so the year before I was born, a great hero in the world of fungal microbial research called Martha Powell did some electron microscopy. So that's microscopy where you can see really fine details of cells. And she saw a structure on the side of these fungal cells that looks just like the eye of insects, the compound eye of insects. And a couple of years ago, we discovered that the proteins that make up that eye structure are similar proteins to what we use to detect light in our eyes. And then we've gone on to show that this structure functions like an eye and enables the fungi to move towards light.
Wow,
that is amazing.
Like rhodopsin or something.
It is a rhodopsin. And what's cool about this rhodopsin is it's fused to an enzyme that generates the messenger that we use to perceive light, to generate an action potential.
Wow.
So sorry, can you break that down for me? Rhodopsin's a protein.
It's a membrane protein.
Okay, so it's on the edge of the cell.
Yeah.
And it gets hit by a bit of light.
Yeah, and there's a conformational, there's a change in shape that occurs. That's the key.
Okay. And so they can see what's going on around them.
Yes. So what's really weird is one subgroup of this fungi has 3 types of this rhodopsin, and it can see from far red light frequencies right through to blue light frequencies. So technically, this fungus can see a wider span of light wavelengths than we can.
Wow.
and respond to it. And it's really, a lot of people are interested in this system because you can manipulate these proteins to generate action potentials directly in cells, so you can stimulate nerves. So people are using them to engineer optogenetic circuits to control nervous responses by shining lights at cells, essentially.
Wow.
So for our listeners, basically, we can send signals.
Yeah.
Is what we're using
you can
these.
flash a light at one cell, and send a signal down a nerve.
And actually, this is how neurobiologists are mapping the brains of rats, essentially, by shining lights on different neurons and seeing how they respond.
My God, well, that's amazing. It sounds like this fungus would go nuts in a disco.
Yeah.
But actually, what we really want to talk about, you've just published congratulations, a paper in a journal called PLOS Biology, which is one of the best journals to publish in if you're a scientist, isn't it, or a biologist?
For sure.
And it potentially cracks something that scientists have been bewildered by for a long time, which is life is either left or right-handed, but not like I'm right-handed.
Well, it's not that dissimilar in some ways. Well, it's not so dissimilar to your hands, shall we say, right? So the concept of chirality is, and this is what we're talking about, we're going to be talking about chirality is, just for our listeners, it's take each one of your hands and your little finger is always connected to your ring finger.
Right, okay, down here, yeah.
And that's always connected to the longest finger.
So they join down.
So both hands will have the same joint.
So if all that fat wasn't there, you can see where my bones were joined.
But they're actually mirror images of each other. You cannot pick up one and superimpose it on the other one without flipping it.
Yeah.
And so certain molecules with certain properties have this effect. So they're connected to the same atoms, but in space, the way they're arranged is different.
And life only uses, so Tom, life only uses one hand.
All the DNA and RNA of life that we know is right-handed, and all the proteins, the amino acids of proteins, apart from one, is left-handed. So, and that one is called glycine, and it doesn't have stereochemistry. So, and the important thing about the reason it's so important for the origin of life is that if you polymerize right, the polymerization can carry on. But if you interrupt that with right, left, then it stops polymerization.
And a polymer is a long molecule that life uses.
So DNA is a amino acids form a polymer to make proteins. So whole of life requires polymerization.
So it has to choose one or the other.
Yeah.
And they're kind of equally abundant, are they?
So if you have an abiotic chemical reaction, one of the reasons it's not produced by life is it's 50% production of right and left-handed. And actually, many of the drugs we use are abiotically produced, and they have a 50% combination of those two things. So,
which is bad.
Which is bad because actually, for example, the thalidomide drug, one stereochemical form stops nausea during pregnancy, and the other stereochemical form causes development abnormalities. So there's been this massive challenge in pharmacology to try and separate the two stereoisomers, and so lots of people think about this, and our discovery that certain membranes act as that is not necessarily new. We've known that there's certain types of compounds will separate stereoisomers, but the demonstration that a membrane that's biologically relevant will do it at very high rates was one of the key discoveries we found.
We should probably just let our listeners know that stereoisomers mean the two different types of molecules.
Yes.
The left and the right.
The left and the right are called stereoisomers.
Yes.
And so if we go back, I guess, in time and in the development of life, at some point, biologically active or actually biologically important molecules became encased in a membrane.
That's right. So we know that's for sure because all life on this planet has a membrane of some type. And what's interesting is that it's not always been the same chemistry. In fact, we have a different membrane chemistry than bacteria do. It's only slightly different. And then archaea have one that's much more different.
And archaea, they're an early microbe that's separated from the bacteria, like.
So in 1977, Carl Woese discovered a third group type of life forms, which are microbial, called archaea. And originally thought they were just a different type of bacteria. But then it was argued that they have so many fundamental differences to bacteria that they were made a separate type of life. And one of those fundamental differences is the membrane chemistry. The phospholipids that make up the membrane are very different to that of bacteria and eukaryotes.
So the eukaryotes and bacteria, in one sense, have similar membranes.
They're very similar, yes.
And then the archaea ones are different. We can argue on whether they're similar or different, but in one aspect that's key for your work, they're similar, right?
Sorry, which one's similar? I lost
track. Sorry, that's my fault. The bacteria and the eukaryotic
are generally
ones,
the same.
similar.
There's one slight difference. And often, all these membranes have additional chemistries added to them, as you well know better than I do. But the archaea ones are very different. Do you want me to explain how?
Yes, please.
So the archaea ones are the chain that forms the lipid. The tail is an isoprenoid chain, whereas in bacteria and eukaryotes it's fatty acids. Then
they're made from different molecules.
Yes, yeah. And then they have a different, and they both have this glycerol phosphate head group. But they are, which is where the
sugary bit.
Yeah, the phosphate joins to the lipid, and that is in archaea. It's bonded at a different point in that sugar molecule, and that bonding reverses the stereochemistry.
So, just for our listeners who aren't familiar with phospholipids and such like, I guess the easiest way to explain it is to say that these molecules have an oil-like fatty acid in bacteria and eukaryotics chain that doesn't like water and excludes it, but of course they're surface facing, so then they also have a component, the phosphate head group and the rest of the bit that goes on top that faces the water and likes to be in water. So they're special molecules. They have a little bit that doesn't like to be in water, and then they also have a bit that likes to be in water. Yeah. That's why they make such good barriers.
Exactly, and it's a combination of those two characters you just described that means that they can self-assemble into these compartments.
So if you take them and chuck them in a load of water, shake them around, they will form a nice...
Generally, yes.
A bit like water and oil.
Yes.
Yeah,
oil
absolutely.
and water.
Yeah, for sure.
Okay.
Yeah, I mean, the classic example is a soap bubble, which is and actually some great experiments showed that you could extract material from meteorites and essentially form these kind of compartments from them. But they were more simplified versions of that. They were made of fatty acids, and they lacked the phosphate bit, which is the bit that attracts water.
Yeah.
And they were less stable, right? But at some point in the evolution of life, we moved to this kind of this fatty substance, this oil that was bound to phosphate, and that gave it this kind of increased integrity, which means that you could form much more stable kind of compartments.
So basically, the types of membranes that you get around bacteria, eukaryotes, and archaea, even though they kind of do differ in their structures a little bit, they quite easily form in water as long as you've got the right chemicals there to get them going.
Yeah,
that's the key.
So yes, they do. There is one. Yes, they do form quite easily, but there's this idea that was a phase before phospholipids, which was just fatty acids, so it didn't have the phosphate, and they will form readily, but they will also
they'll break apart.
Much more easier, especially when they're in when there's ions around. And actually, the ions are quite important because they drive some of the chemistry that enables polymerization. So you need a step beyond that where the lipid becomes a phospholipid and becomes more stable.
Cool. And so at some point, these molecules surrounded and formed a barrier and encased and protected.
Yes.
Other biologically important molecules.
Yeah, so that was there was a theory for that called the Obell Hypothesis, which is essentially that kind of bag kind of encapsulated some biochemistry that was occurring, and therefore enabled it to become subdivided from the environment around it.
So like a proto-cell.
Yes, yeah.
And so your latest findings suggest that it might be, I mean, I'll let you describe it to us, but something about the properties of the membrane itself had something to do with what was encased inside.
I was wondering why where the left-handed, right-handedness was going to come in.
So there's been lots of study on the bacterial type and the eukaryote type membrane chemistries we talked about earlier, but really the archaeal type ones have been ignored. And so our study was to focus on what is the permeability characteristics of archaeal membranes. And that's when we found out that actually in many cases they're much more permeable. And actually some of that permeability characteristic selects for certain types of stereochemical isomer. So the key initial finding was that the archaeal membrane would exclude left-handed, must get it right when it excludes the left-handed ribose and deoxyribose, but and therefore allow much higher rate of uptake of the right-handed deoxyribose and ribose. And they're the pentose sugars which are so critical for forming DNA and RNA.
Please, if you're enjoying this, do subscribe, do like, and please share it. We'll be back in a minute.
Hi, Tim. You're looking particularly snazzy today. What are you wearing?
I'm wearing a piece from a company called Grace and Garbo. And actually, you know a little bit more about them than I do, to be honest.
Yeah, you're right, I do. And
you're the fashion queen. Yeah.
I'm also wearing a piece from Grace and Garbo. So this is a delightful knitwear outfit set up in Birmingham. What they do is they source knitwear, quality knitwear from all around the world. So it's vintage, one-off pieces, many of which they then hand embellish, like the one you're wearing today with its beautiful purple flowers. And so they're big on sustainability. Nothing goes to waste. Everything is sort of recycled and reused. And if you want to know more about them, then have a look at the link beneath this episode.
And thank you very much to them for supporting our podcast. Hope their sales soar.
Yeah, thanks a lot, Grace and Garbo.
Welcome back, everyone. And we're talking to Tom Richards about why life favours one-handed, right-handed or left-handed, depending on whether you're talking about the RNA and DNA or the protein, the amino acids and the proteins. And so there's obviously quite a lot to talk about because in the break, which we may put a little discussion of, the two chemists, the two people who know more about chemistry than me.
I'm definitely not a chemist.
Yeah. We're going at it anyway.
in a friendly way,
in
right?
A friendly way, yeah.
Yeah, so it's important to recognise that all the model systems we're working on are one specific type of phospholipid and highly pure. I mean, which is not very biologically realistic, but that was chosen on purpose because we really wanted to understand the characteristics of certain types of phospholipids. Yeah.
And you have the most, PC is the most abundant one in eukaryotes, so that makes sense.
Yeah.
So what did you do? What was the experiment?
I have to say, this is all a collaboration with my great friend Stefano Pagliara, who's at the University of Exeter. He's a physicist, and he spent a lot of time studying how chemicals cross membranes, much like yourself, Simon. And what we were trying to do is recreate kind of membrane chemicals which are alternative which might have been used at alternative phases of the tree of life. And so what you do is you take this mixture of phospholipids and you electroporate it, and they'll form these big vesicles, so these big spheres. So they're forming a compartment, and these are 10 microns in size. So, roughly equivalent to many cells that...
Is that like a bacteria cell? Oh, sorry, eukaryote cell?
Yeah, that's a most... small eukaryotes about 5 microns.
Okay.
Yeah, so
you take your electrodes, right? You take your electrodes, you put them on the membranes. Yeah, that's the electroporation part, right?
Yeah, and it forms it, and then we run these spheres, these tiny spheres. We have this kind of we've built these unique type of plumbing devices called microfluidics, and we put those spheres and we capture thousands of those spheres in little compartments so we can look at them down a microscope, right? And because they're in this plumbing device, you can control the liquid that's moving through that microfluidic device, and then you can change what chemicals are outside the membrane. And before we put those membrane vesicles in the microfluidic device, in this plumbing device. We fill them with a fluorescent reporter, and that fluorescent reporter gets more fluorescent if it's in contact with compounds which have crossed the membrane into the vesicle, the spherical vesicle.
That's really cool. So basically, you've got these round balls.
Yeah.
You fill them with some fluorescent dye, which fluoresces even more once the molecules go in, and that gives you your signal.
Yes.
That's pretty neat.
And then you turn the tap on the plumbing device, and you run through a different a buffer, a fluid that's got a different compound in it, and you can see how much of the compound crosses the membrane by monitoring how the fluorescence changes.
Is the color always the same for the different molecules?
Yes, it is. So you have to do them separate. You have to do them separately, one at a time.
Okay, so you do them one at a time. You can see that the left-handed ones are being taken up more quickly than the right.
Yes. So that's something we're interested in, right? Is if you had a 50-50 mix, would it change the rate of uptake? And we've been doing some very preliminary molecular dynamic simulations, which I'm always happy to get advice on. And one of the things we look like is that certain types of the stereochemical form seems like they stick to the top of the glycerol phosphate, the top of the phospholipid, and that might be affecting how permeability progresses. So that's something we're interested in doing long term. So that's me trying to elicit a collaboration out of this.
No,
that'd be great.
Yeah.
And so, has it cracked the problem of why life, is kind of right-handed for the DNA and RNA and left-handed for the amino acids?
So it provides an explanation for why it's adopted, but there is, as scientists, we have to criticize our own work. And the main criticism of this work is that the type of phospholipid we're using, there's very little fossil record for that type of phospholipid deep in. Is
that right?
Yeah.
For PC?
No, for the isoprenoid.
Yes, right.
Yeah, type. Yeah.
But would it fossilize?
So you're right. No, you can get chemical traces from from fossil, but you're right. But there is for PC, but not for the. So geochemists are skeptical from that perspective, right? Now there's lots of ways you could imagine that if there's a bit that the process of vesicle growth, which we can talk about a bit more if you're interested in, would actually lead to purified vesicles, which could do this, and therefore you get a system which is selecting its own growth, so it becomes like a Darwinian, it's under selection system, but it's still, there's not much evidence that this type of phospholipid chemistry was present early in abiotic conditions, which means that it had to have probably come about through metabolism, which means you would have had to have some kind of proto-life form before this type of membrane was utilized. So it's possible that this is, like the second act in evolution of life.
Rather than the very first.
Yeah.
It's quite interesting, actually, because one of the very first podcasts we did was we talked to Adi Pross.
That's right.
And we were talking about some of these issues and, the origin of life. And we also, we were talking, just talking about sort of the self-propagating, like you said, the selection rules and such
Yeah.
And it's interesting how it
all fits together.
Yeah.
So the reason we're so excited about this is because actually, if you get polymerization, like I was talking about, with right on right on right for DNA and RNA and left on left on left for amino acids, inside a vesicle, a protocell, that vesicle, if there's enough phospholipid hanging around, will grow.
Right.
Okay.
Right.
And as it grows, the surface area increases. Yeah. As the surface area increases, more permeability function occurs.
Okay. So you get more into the cell.
And you get what you'll understand, Tim, as like a Darwinian demon.
Yeah.
It's much more competitive than the things around it because you have a reinforce it. You have a circular economy. And that's why we were so excited about this. Yeah.
No, it's very cool. I mean, there's very few rocks that are 4 billion years old, right? When this would have got going. So is the lack of kind of fossil evidence really that damning?
I don't think so, but I would say that. So, so it comes back to these meteorites. So most of the evidence, because most of the rocks on Earth are full of contamination, essentially. But when you study meteorites and you look at the chemical composition of meteorites, you can see like fatty acids come out of it, which is the main components of bacterial membranes, not archaeal membranes. And we were studying mainly archaeal membranes.
Okay.
Now, there's a Japanese group that said you can get some selection for stereochemical compounds over bacterial type membranes, but they showed it over a process of 15 hours. So it's very slow.
Very, very slow.
And our process was over 3 minutes.
Yeah,
So
sure.
it was much more significant.
So, and we also spoke to Sarah Russell.
We did,
didn't we? Who was in charge of the OSIRIX-REx. Was involved in the OSIRIX-REx mission that went to Bennu and did this touch and go and grabbed some asteroid material, brought it back to Earth, and they analysed that and found, 50-50.
Yeah, she was looking at the sort of more inorganic.
Yeah, she was looking at
but I spent a lot of time going through their supplementary material, looking at what amino acids are in there and where do we see selection across membranes for those amino acids. And what was really cool about that study is it was one of the first pieces of evidence I certainly saw where there was an amino acid called arginine being produced, which I didn't think would form abiotically because it's such a big amino acid. And it was there, not huge amounts, but it was there.
Wow.
And arginine was one of the ones that showed the biggest selection for left-handed permeability, along with alanine. So you've got one of the biggest ones, arginine, and one of the smallest one, alanine.
That's amazing.
So I was quite excited by that. So that I was really excited by those papers.
Well, what I'm really impressed at is, every time we write a paper, our listeners, not all of our listeners will know this, you put a lot of the detail into supplementary information, and I don't think I've ever read a supplementary information, so I'm very impressed, Tom.
Well, it's one of the few occasions I did because it was such an exciting result, to get that data so clean.
I'm going to ask what I think is probably likely a very dumb question.
Okay.
And especially given I'm supposed to know something about this area.
Okay.
So, do we know which way round things went? So, could it be that the permeability... Do you know what I'm trying to say?
Yeah.
Could it be that the permeability came first and then we had the encapsulation? Or could it be that those were the molecules that needed to be encapsulated and so the lipids were...
Yes, so it's a bit of a chicken and egg question, right? And that's a lot of the origin of life research is like that. So we don't know which way around it is. In the paper, there's a section where we say that actually either way round, having this permeability selection would be favourable. So if you're evolving life from scratch inside a membrane, then you would be able to assemble the components which would favour polymerization. But then if you had a system that's membrane free, it would be constantly inhibited in terms of function by contamination from the wrong stereochemistry.
Yes.
It may possibly have been able to have occurred, but then moving to this membrane would immediately give it an advantage over stuff that's not. Now, there's some cool evidence coming out that actually the steps in ribosynthesis, I'm going to get this wrong, might get this wrong, have certain magnetic property. When they involve an ion, when they're catalyzed by ion, in cell-free abiotic chemistry, you can get different enantiomers, stereoisomers are either attracted or repulsed, and so there is a way that in certain environment conditions you can get
you can separate them
subunits so you could have a step where the compounds are forming abiotically that life would require, and then but to really to increase its rate of production, it needs to move into an environment where it excludes those other stereochemical isomers which interfere with it, and that's why we think the membrane is important.
That is so cool. So I read a report, actually, no, I'm exaggerating massively. I read a news article about a report that came out recently that said there is an existential threat to life on Earth if chemists were to create life that's the other-handedness. So left-handed DNA and RNA and right-handed right-handed amino acids. And if they did that, apparently it would, be a massive bioweapon, and we'd all be at risk of immediate death. Did you hear about this report?
I did read about it. So this is the kind of mirrored life
That's
report.
Right, mirrored life, yeah. Is that nonsense, or should I be lying awake at night worrying?
It's one of those things that you could imagine in ways it could be devastating, but whether they're actually realistic or not is difficult to imagine. I mean, that's quite a circular statement. But
I know what you mean, though. Yeah.
So let me think about this. So I think the main fear is that essentially you would produce agents that are kind of undetected by cellular process, that they can interfere with physiology. And
so a bit like the developmental stuff you were talking about with the thalidomide.
Yes. Yeah, essentially, yeah.
Okay.
And things like that. So there's lots of unintended consequences like that. But in terms of the core function, I can't quite imagine. I mean, other than because if these things are being packaged up in polymers separately, then they're not interfering with each other. It's when you mix the compounds
together, that's when the trouble starts.
Yeah, I think I agree with that. Also, now, fundamental reactions are the enzymes for those are so optimized. Yeah. They probably they recognize the wrong one, probably just ignore it, maybe. I don't know.
I agree. I think they would ignore it.
We're running out of time, but I've got one more question, if I may. Do you think we're getting close to being able to make artificial life in the laboratory? I mean, you've talked, as you say, about, why it uses kind of one-handedness over the other. This guy, Adi Pross, was talking about dynamic kinetic states, and so basically turnovers.
Yes.
We've talked about membranes here forming.
And David Baker was also talking about design
of proteins. I mean, are we close, or is it a long way off? What are your...
I think you could take it, if you take not just our work, lots of other work about this idea that you can make self-sustaining polymerization inside vesicles. You could make a system that looks like a living organism, as in it's like growing and replicating, right? But it's not doing metabolism to sustain itself. Okay. It's requiring an environment which provides all the metabolites in place. Now, there's lots of life forms that are like that, but it has some characteristics of life. So I think you could do that, but whether it would be so fragile that it's not really a stable organism.
So it wouldn't be able to make and use its own energy. I mean, you can't make energy, but you know what I mean. So
I know what you mean. Yeah. No, it wouldn't be able to do that.
Right.
No.
Well, Tom, thank you very much for coming on. Congratulations on the paper. It's a really great bit of work. For
sure.
And I love listening about that. And we'll have to get you back on with the next breakthrough at some point.
About the eyes. I want to know about
You want to know about the fungi?
Yeah, the fungi with the eyes.
Yeah. No, fantastic.
Thank you for having me.
Well, that was absolutely fascinating, Simon. Honestly, I had no idea that chemistry could be so interesting.
I don't know why I put up with you. No, absolutely. I think it's opened up, it's answered lots of questions perhaps, but it's also opened up lots of questions. And so I think that's what's also really nice about it, is that Tom, he tells it like it is. And he's explained that, these membranes that they've worked with are simplified versions of the real membranes, and it leaves a lot of other questions. I think it's going to be an interesting thing for us to keep an eye on for a long time.
No, I think one of the things this is what science so often does, right? You make progress on one problem, and then it throws up a load more questions, right?
Absolutely. And it has to, right? None of the questions we address there's never a full stop at the end. There's always something else. And I think...
Which is just as well, otherwise we'd do ourselves out of a job every time we...
You'd be on holiday every day, Tim.
Something like that. But no, I think it's fascinating stuff, and let's keep an eye on it. And you might have got a new collaboration by the sound of it.
Yeah, well, I think Tom and I have been trying to do this for a while, so we might get around to it now.
Yeah.
Absolutely.
Well, if you want to know more, go and read the paper, which is in the link below, and please subscribe, and please like, and please share on social media. And we'll see you next week when dinosaurs, perhaps?
Possibly. Yeah, who knows? Could be dinosaurs, could be wasps, it could be anything. You'll just have to watch out, folks.
We're more organised than that, listeners. Okay. Goodbye.
Bye.